Hi everyone, my name is Ayelet. I'm a student at Gordiar's lab, and with me, Or will also be presenting. So we'll talk about today on IG germline variations. So first of all, a little background. So the germline variation or B cell diversity can come from two levels. We have the first one, the genetic levels, which we usually find in naive B cells, where we have the VGJ rearrangement, the DNA rearrangement. And the second level is the more environmental, which the affinity maturation. And what we wanted to explore here basically was the variation or the diversity within the chromosome, within the IG loci. So for that, we usually go to the B cell repertoires, or in our case, the naive B cell repertoire. And to explore that, you go to see what there is in the chromosome. So the IG chromosome is quite hard to sequence due to a lot of repetitive sequences within it. And also it's quite hard to map it back to the full chromosome. So using the Immcantation tools and the data sets we acquired, we try to complete the picture and see what genes and alleles we have in the chromosomes, and also how do they align on each of the parental chromosomes. So for that, we collected a data set of 100 individuals, and we sequenced the repertoire of their naive B cells. And we have both the heavy and the light chains from all individuals. And about half of the population has celiac disease. So first of all, we used the pre-processing pipeline of Immcantation. And one major step for us was using Tiger. So we used it in two different ways. First of all, we used it to find novel alleles. So in our data set, we found 26 novel alleles. And just recently, we our partners published a paper about the 26 novel alleles. And the other side is that we used it to infer Bayesian genotype. Basically, the genotype meaning which genes and allele an individual had. And what we like most about this new genotype inference is the confidence level that we acquire from using Bayesian statistics. So here on the right is just a small representation of a genotype map where each row represents a gene. Each color here on the right represents a different allele. And the column here on the left represent our confidence level in each genotype inference. Now, knowing the exact alleles and genes of an individual can help downstream analysis. When we have misassignments of each, it can introduce a lot of bias that will eventually influence the decision that we will make. So this was an important step that we used Immcantation tools. And it helped us a lot to later on look at the gene distribution. So once we knew exactly what genes and allele we had, it was easier to look at the gene distribution of all the individual in our data set. And what we saw is that there are overall a consistent picture of the gene usage for all individuals, but in some cases, in some genes, we see that there is sort of like a bimodal distributions. And we can see it, for example, in V39. Also, we can see it here in V438 too, where you can see a clear separation where one population is found on 0 and the other one is relatively high to it. Now, each dot here represents the relative gene usage of one individual. And what we were mostly interested about is why do we see this clear separation and what does it represent? So what we did is we used a binomial test to basically ask how far is the lower population that we see here from the upper one. And if it was far enough, and if it was far enough, then we said that probably this area, this group of individuals don't show this gene, or there is a problem with transcribing this gene. So either it was deleted from the chromosome, or there is a different issue. So those genes were suspected to be deleted. And what we did next from that is going back to the chromosome and sequencing the area of this gene to see if it's in fact deleted. And what we saw is that for some of the genes that were suspected to be deleted, they are in fact deleted from the chromosome. So this allows us to add another layer of variation that we didn't think of before. And what I'm going to show now is the complete map of it. It's an extension of the graph that we saw before. On the top, we have the gene distribution. Each row here on the bottom represents a different individual, where the color blue represents a deletion, and the white represent no deletion, and the dark gray represent a decision of unknown. Those did not pass the binomial test with a high confidence level. And what's interesting here is seeing the different patterns, the different variations that we see. For example, here we can see that there are two genes that are always deleted together. And if they're deleted together, then those two genes are not deleted, and vice versa. And not only that, there are also another couple of genes that are deleted together. So this added another layer and show that there are patterns within the chromosome when we look at the deleted area, and not only at the usage. Also, a lack usage is a variation. So building on that and all the Immcantation tools, we developed Rabbit, which is an extension to the Immcantation. And Rabbit comes to infer haplotype from B-cell repertoire. So as I said before, it's quite hard to build back the chromosomes from small fragments. But if we use a repertoire where there is at least one heterozygous gene, we can get the haplotypes, the parental chromosomes. So Rabbit, what it does, it takes one heterozygous gene. It can be either J6 gene, which is widely known to be heterozygous, or it can take also one D gene that is heterozygous, for example, D28 or D221. and recreate the chromosomes. So what we see here on the left is a single haplotype map where the most left panel, where each row represents a gene, the color represents the different alleles, and dark gray represent here a deletion, and the blues represent our confidence level. Here we also used what Tiger started with Bayesian inference. We used it also here. We did a haplotype based on Bayesian inference. Now this left panel represents the count that we have from each gene recombined, in this example for J6. The middle panel show the inference we have, the decision of the inference of the allele for each chromosome, and the right panel show the confidence level. And here, what we did, we added another visualization that help you compare between different individuals and their chromosome to try and search if there is another connection between what we see in one individual to another based on the haplotype inference. What is interesting with this Bayesian inference is that it allows us to detect single chromosome deletions. As we can see here, all the gray boxes represent a deletion. Now the top panel represent the J602 chromosomes, and the lower one represent the J603 chromosomes. And what we saw is that in some cases, a gene can be deleted only on a single chromosome, not on both. For example, here. We did that for the entire population, and what we can see here that we got a map of the single chromosome deletions. Now, a single chromosome deletion in rabbit is defined as basically an unknown reference with a high confidence level. So here, each row represents a single individual. The top is the J602 chromosome, and the lower is the J603 chromosome. The dark color of the blue represents a deletion with high confidence level, while the light blue represent an unknown inference with a low confidence level. The gray is an unknown inference with a high confidence level, and the white is no deletion. Now the top panel represent the sum of the deletions. And what's interesting here is, again, we see patterns of the deletions with coupled genes. So again, we see that V39 and V18 are deleted together. And while they're deleted, V5101 and V364D are not deleted, and vice versa. The very interesting thing that we saw here is that for one individual, we have a stretch of about 100 kilobase pairs that are deleted from the chromosome. Until now, we were unable to understand where it came from, or to what exactly it's attached to, or what it influenced. This individual does not have the celiac disease, so we know it's not connected to that. But what we hope to see is, if this is used in many other individuals, then maybe this occurrence happens again, and we'll know the connection from there. So this and the other variation let us think that if we enlarge our database and recreate what we did now on a large scale, maybe we can get more information and understand better on the variations and see the different patterns that we saw in 100 individuals. And for that, we developed VDJ base.
Okay. So our motivation to explore the genetic immunoglobulin Germline variation led us to develop a new database application that stores the genotype and haplotype data inferred from published AIRR-seq datasets. So VDJ base allows the scientific community to study the genetic variability and compare between different samples. To date, the site contains more than 500 samples from different 14 projects, but not all the projects were done by the same sequencing or the same libraries. So in order to be able to compare different samples, we have developed a uniform pre-processing pipeline that all the samples must pass before entering to VDJ database. In addition, for easy comparative data visualization, we develop two R packages, VDJ Viz and Rabbit. All of these tools are the basis for VDJ base, and all codes are available in Bitbucket. Okay, so let's look on a snapshot of search section page of VDJ base. Our applications allows users to query and filter data according to selectable metadata criteria, such as clinical diagnosis, ethnic group, gender, cell type, and etc. The results are displayed as a table where each row represents a sample along with their rated metadata, such as organism, target locus, heavy or light chain, total reads of this sequencing sample, cell type like PBMC or naive cells, and these two columns represent the results the files that we create from the Tiger. So here we can see the files that can be freely downloaded, and we have a two representative visualization for one individual. In addition, for graphs for comparative samples, we have this left side bar that enable the creation of visual analysis for users' selected samples. Using these advanced filters, this is a set of a drop-down tag list. A user can filter all the visualization according to specific genes, alleles, or certainty levels of inference for each genotype or haplotype decision. For more details on how to use it, you can find the tutorial videos on user guide page. So as I mentioned, our database includes online tools to assist in visualization and to help users gain insight into the data. So these are some examples of visualization you can create in the database. A represents a compare multiple genotypes, that each row represents a sample and each column represents a gene. Colors represent different alleles. And on the same way, you can compare multiple haplotypes as Ayelet continues. You can see in C heterozygosity abundance for each gene in the samples of interest. And in D, you can see gene usage. Here, each point represents an individual, and color correspond to V family gene. What is interesting thing here is that we can see that we see the same result that Ayelet see in the 100 celiac data, and this is on all the studies we have in the database. So we see the two groups for V3 and 9. Another graph that we can create is the allele appearance, allele distribution, that the left y-axis correspond to the number of individuals, and the right y-axis correspond to the frequency of the allele in the samples of interest. Okay, so our future plans is to adapting Tiger rabbit and VDJ base to other species and to T-cell receptor data, adding more projects to VDJ base, adding and improving comparative visualizations, and creating an interface between different servers and VDJ base. And as we like to say, and just use it. And I want to thank the Immcantation teams and to all the groups of Go Yari Lab. Thank you.